Hi, I am Dr. P.R. Padma, Professor in the Department of Biochemistry, Biotechnology and Bioinformatics at Avinashalingam University for Women, Coimbatore. You have now chosen to study about the components and structure of nucleic acids, which is a module in the Nutritional Biochemistry paper of the subject Food Science Nutrition. Nucleic acids play a central role in the functioning of every cell. They have the ability to store and transmit genetic information that governs the function of the entire cell and thus the entire organism as well. It is very vital for every student of science to understand the structure of nucleic acid and their components. There are two types of nucleic acids in the cells. They are deoxyribonucleic acid, otherwise popularly called DNA, and ribonucleic acid, popularly known as RNA. Both these are polymers of nucleotides. This means they are made up of many units called nucleotides. In other words, the monomers of nucleic acids are nucleotides. Nucleotides themselves have three major components, namely a pentose sugar, a nitrogenous base, and a phosphate group. Nucleotides themselves have a variety of roles in cellular metabolism apart from being the monomers of nucleic acids. They are the energy currency in the metabolic transactions, the essential chemical links in the responses of cells to hormones and other extracellular stimuli, and the structural components of an array of enzyme cofactors and metabolic intermediates. Apart from these, they are also constituents of nucleic acids, both DNA and RNA. Before going into the details of the components and structure of nucleic acids, let us understand the objectives of this module. After completing this module, you will be able to comprehend the differences between the two types of nucleic acids, that is DNA and RNA, the major subtypes of RNA, the components of nucleic acids, and the structure of nucleic acids. Let us start with the structural components of nucleic acids. As we mentioned earlier, the nucleic acids are polymers of nucleotides, and the nucleotides themselves contain three components, namely the pentose sugar, nitrogenous base, and the phosphate group, which forms the phosphodiester bond that links two nucleotides together. We shall take these up one by one. Let us start with the sugar component. As mentioned earlier, the sugar present in nucleic acids is a pentose sugar. This means that the sugar contains 5 carbon atoms in its ring structure. The type of sugar present in RNA is ribose, hence the name ribonucleic acid or ribonucleic acid. This figure shows the structure of ribose. In the sugar, the carbon atoms are numbered from 1 to 5, and to differentiate them from the carbons in the nitrogenous base, they are marked with an apostrophe sign, which is referred to as a prime. Thus, the ribose has 5 carbons from 1 prime to 5 prime. DNA also contains the same sugar, but with a major modification in its structure. That is, on the second carbon of the sugar or on the 2 prime carbon, one oxygen atom is removed. Hence, the sugar is called a 2 prime deoxyribose. The structure of this sugar is shown here. Because of this, the nucleic acid containing this type of sugar is called deoxyribonucleic acid or DNA. Thus, the first major difference between RNA and DNA is the sugar. Both are pentoses, both are ribose sugars, but in DNA, this ribose has a deoxygenated carbon at the 2 prime position. Now, let us move on to the next major component, namely the nitrogenous bases. As the name implies, these components are basic in nature and contain nitrogen as an important part in the ring structure. There are two types of nitrogenous bases, namely the purines and the pyrimidines. Both of them are ring structures. Purines contain two rings fused together, while pyrimidines contain a single ring structure. The structure of purine and pyrimidine ring structures are shown here. Now, you will see that the atoms in the ring are numbered sequentially, but without the prime sign. This is to differentiate them from the atoms in the sugar. Both DNA and RNA contain two major purine bases and two major pyrimidine bases. The purine bases in both DNA and RNA are the same, namely adenine, shortly denoted as capital A, and guanine as capital G. In both DNA and RNA, one of the pyrimidines is cytosine, denoted by a capital C. But the second major pyrimidine is not the same in DNA and RNA. It is thymine in DNA and uracil in RNA, denoted by capital T and capital U respectively. Very rarely, thymine occurs in RNA and uracil in DNA as well. The most important functional groups of pyrimidines and purines are the ring nitrogens, carbonyl groups, and exocyclic amino groups. These groups are vital in forming the hydrogen bonds that hold the strands of nucleic acids together. Hydrogen bonds involving the amino and carbonyl groups are an important mode of interaction between the bases in nucleic acid molecules. Hydrogen bonds between bases permit a complementary association of the two strands of nucleic acid. The most important hydrogen bonding pattern are those defined by James Watson and Francis Crick in 1953, in which A bonds specifically to T in DNA or U in RNA, and G bonds specifically to C. These two types of base pairs predominate in the double-stranded DNA and double-stranded RNA. Each nucleotide base of one strand is paired in the same plane with the base of the other strand. In the double-stranded DNA or RNA, the number of adenine bases equals the number of thymine or uracil bases, and the number of guanine bases equals that of cytosine. The structure that is formed by the combination of the sugar moiety and the nitrogenous base is referred to as a nucleoside. Thus, we have ribonucleosides which contain adenine, guanine, cytosine and uracil, and the deoxyribonucleosides which contain adenine, guanine, cytosine and thymine. The third component of the nucleotide is a phosphate group. This phosphate group attaches to the hydroxyl group on the 5' carbon of the sugar moiety. This results in the formation of an ester bond, because ester bonds are formed when hydroxyl groups react with acid groups, which in this case is phosphoric acid. Thus, the general structure of the nucleotide monomer can be represented as shown in this figure. The nitrogenous base is attached to the first carbon, the 1' carbon, and the phosphate group to the 5th carbon or the 5' carbon of the sugar. Thus, the nucleotides are formed by the combination of a phosphate group to nucleosides. Having understood the structural components of the monomeric units of DNA and RNA, now let us consider how these units are linked together to form the polymers. For forming the polymeric structures, the nucleotides are linked to each other by a reaction between the phosphate group of one nucleotide and the next nucleotide. In this reaction, one more ester bond is formed, that is between the phosphoric acid or phosphate group and the 3' hydroxyl group. Thus, the phosphate group is flanked on both sides by an ester bond. This bridge is therefore referred to as a phosphodiester bond. Therefore, the nucleic acid strands are formed by stringing together nucleotide monomers which are linked to each other by phosphodiester bonds. These phosphodiester bonds form the backbone of the nucleic acid strands. The sequence in which the nucleotides are strung together is extremely vital and is responsible for the genetic information that is stored and transmitted in the cells. Any mistake or damage in this sequence can lead to complications in the functioning of the cells and also to disease conditions. The two strands of double-stranded DNA helix are held together by hydrogen bonds that are formed between the purine and pyrimidine bases of the respective linear molecule. The pairings between the purine and pyrimidine nucleotides on the opposite strands are very specific and are dependent upon the hydrogen bonding of A with T and G with C. The two strands of the double helical molecule, each of which possesses a polarity, are anti-parallel to one strand running from 5' to 3' direction and the other in 3' to 5' direction. This figure shows the schematic representation of the DNA structure. The formation of this helical structure results in the formation of depressions in the structure. These are called grooves. The winding of the helix results in the formation of deeper grooves called major grooves and shallower grooves called the minor grooves. In the normal configuration of DNA, which is called the B-DNA, the distance between one major groove and the next is 34 angstrom and spans 10.5 nucleotides. Let us now summarize the key features of the structure that we have learned so far before moving on to the other features. The monomeric units of nucleic acid polymers are nucleotides, which are in turn made up of pentose sugars, nitrogenous bases and phosphate groups. The monomers are linked together by 5' 3' phosphodiester bonds. The two strands are held together by hydrogen bonds formed by complementarity of base pairs. The AT pair has 2 hydrogen bonds while the GC pair has 3 hydrogen bonds. Thus, the GC pair is stronger than the AT pair. The complementary base pairing in DNA helix proves Chargaff's rule, that is, the contents of adenine equals the content of thymine and guanine equals that of cytosine. The two strands run anti-parallel to each other, which means one strand runs in the 3' to 5' direction while the other runs in the opposite or 5' to 3' direction. The sequence of bases governs the genetic information in the DNA molecule and the transmission of this information by the RNA molecule. The genetic information resides on one of the two strands, which is known as the template strand or sense strand. The opposite strand is called the anti-sense strand. The double helix has major grooves and minor grooves along the phosphodiester backbone. Now, let us consider the other features of the double helix. Variations in the conformation of nucleotides of DNA are associated with conformational variants of DNA. The double helical structure of DNA exists in at least 6 different forms, namely A to E and Z or commonly Z. Among these, the B, A and Z forms are the most important forms of DNA. The B form of DNA double helix described by Watson and Crick is the most predominant form under physiological conditions. In this form, each turn of the helix has 10.5 base pairs spanning a distance of 34 angstrom units. The width of the double helix is 2 angstrom units. Structural variations in DNA reflects 3 aspects, namely the different possible conformation of the deoxyribose, the rotation about the contiguous bonds that make up the phosphodiester backbone and the free rotation about the C1N glycosyl bond. The Watson and Crick structure is also referred to as the B form of DNA or bDNA. The B form, as we mentioned, is the most stable structure for a random sequence. DNA molecule under physiological conditions is in the B form and is therefore the standard point of reference for the structural properties of DNA. 2 other structural variants of DNA that have been well characterized in crystal structures are the A form and the Z form. The A form is favored in many solutions that are relatively devoid of water. The DNA is still arranged in a right-handed double helix as was the bDNA, but the helix is wider and the number of base pairs per helical turn is 11 rather than 10.5 as in bDNA. The plane of the base pairs in aDNA is tilted by about 20 degrees with respect to the helix axis. These structural changes or deepen the major grooves while making the minor grooves shallower. The reagents used to promote crystallization of DNA tend to dehydrate the DNA, and thus most short DNA molecules tend to crystallize in the A form. The Z form of DNA is a more radical departure from the B structure in that the most obvious distinction being that the Z DNA is a left-handed helical rotation. There are 12 base pairs per helical turn and the structure appears more slender and elongated than bDNA. The DNA backbone also takes on a zigzag appearance in the Z conformation. Certain nucleotide sequences fold into the left-handed Z helices much more readily than the others. Now let us move on to the types of RNA. RNAs have a broader range of function and several classes of RNA are found in the cells. The major types among them are ribosomal RNA or rRNA, messenger RNA or mRNA, and transfer RNA or tRNA. Ribosomal RNAs or rRNAs, as we mentioned, are components of ribosomes, the complexes that carry out protein synthesis. RNA has no simple regular secondary structure that serves as a reference point unlike the double helix of DNA. The three-dimensional structure of many RNAs, like those of proteins and peptides, are complex and unique. Weak interactions, especially base stacking interactions, play a major role in stabilizing RNA structures just as they do in DNA. Where complementary sequences are present, the predominant double-stranded structure is an A-form right-handed double helix. Messenger RNAs or mRNAs are intermediaries carrying genetic information from one or a few genes to a ribosome where the corresponding proteins can be synthesized. The mRNAs are complementary copies of the sense strand of DNA. The mRNA is synthesized in the nucleus in the eukaryotes as heterogeneous nuclear RNA or hnRNA. hnRNA undergoes another process called splicing, wherein the intron or non-coding sequences are removed and the exons or the coding sequences are strung together. Splicing results in the formation of the final functional mRNA molecule. This is then stabilized by the addition of small but specific sequences of nucleotides on both ends, which form a cap on one end and a tail on the other end. The eukaryotic mRNA is capped at the 5' terminal end by 7-methylguanosine triphosphate, which forms the cap. It is believed that this cap helps to prevent the hydrolysis of mRNA by 5' exonucleases. The tail is usually made up of several adenine nucleotides and is therefore called a poly-A tail. This tail may provide stability to mRNA besides preventing it from the attack of 3' exonucleases. The functional mRNA then enters the cytoplasm to participate in protein synthesis. Although mRNAs from different genes can vary greatly in length, the mRNAs from a particular gene generally has a defined size. The process of forming mRNA or any RNA for that matter from a DNA template is known as transcription. mRNA has a high molecular weight with a short half-life. The third type of RNA is the tRNA or transfer RNA. These are adapter molecules that faithfully translate the information in mRNA into the specific sequence of amino acids. Although RNA has certain similarities with DNA structure, they have specific differences. Namely, the sugar in RNA is ribose in contrast to deoxyribose in DNA. RNA contains the pyrimidine uracil in place of thymine in DNA. RNA is usually a single-stranded polynucleotide. However, this strand can fold in certain places to give a double-stranded structure locally if complementary base pairs are in close proximity. Due to the single-stranded nature, there is no specific relation between the number of purines, pyrimidines that are present in RNA. Thus, there is no following of Chargaff's rule. Alkali can readily hydrolyze RNA to 2'3' cyclic diesters. This is possible because of the presence of the hydroxyl group in the 2' position. DNA does not have this oxy molecule over there and therefore cannot be subjected to alkali hydrolysis. It is therefore a more stable molecule. RNAs can be histologically identified by the arsenol color reaction due to the presence of ribose. There are at least 20 species of transfer RNAs corresponding to 20 amino acids present in the protein structure. The structure of tRNA was first elucidated by Hawley. tRNA contains mainly 4 arms, each arm with a base paired stem. They are the acceptor arm, the anticodon arm, the D arm and the T psi C arm. All the tRNA molecules contain the 4 main arms. The acceptor arm is capped with the sequence CCA from the 5' to 3' direction and the amino acid is attached to the acceptor arm. The anticodon arm with the 3 specific nucleotide bases, namely anticodon, is responsible for the recognition of the triplet codon on the mRNA. The codon and anticodon are complementary to each other. The D arm is so named due to the presence of dihydrouridine. The T psi C arm contains a sequence of T, pseudouridine represented by psi and C, that is cytosine. There is additionally a variable arm in tRNA. As the name implies, this is the most variable sequence in the nucleotide sequence of RNA. Based on this variability, tRNAs are classified into 2 categories, namely class 1 tRNAs and class 2 tRNAs. The structure of tRNA is shown in this figure that is now displayed on your screen, where you can see the 4 arms and the variable arm also. Ribosomes are the factories of protein synthesis. The eukaryotic ribosomes are composed of 2 major nucleoprotein complexes, 60S and 40S subunits. The 60S subunit contains 20S rRNA, 5S rRNA and 5.8S rRNA, while the 40S subunit contains 18S rRNA in the eukaryotes. The function of rRNAs in ribosomes is not clearly known. It is believed that they play a significant role in binding of mRNA to ribosomes during protein synthesis. There are several other subtypes of RNA. Among these, the most important ones are the catalytic RNAs, otherwise called ribozymes, and the siRNA or small interfering RNA. Ribozymes or the catalytic RNA are responsible for splicing of hnRNA into mRNA. These are the only non-protein enzymes known. The siRNA causes the degradation of specific mRNA sequences in the cells, resulting in post-transcriptional gene silencing. This process is also called RNA interference or RNAi. This has several applications as a research tool and has fetched the Nobel Prize to the scientists who discovered the process. Let us now summarize the salient points of this lesson that we have gone through. There are 2 types of nucleic acids, RNA and DNA. The nucleotides in RNA contain ribose and the common pyrimidine bases are uracil and cytosine. In DNA, the nucleotides contain 2-oxy-deoxyribose and common pyrimidine bases are thymine and cytosine. The The primary purines are adenine and guanine in both DNA and RNA. Native DNA consists of two anti-parallel chains in a right-handed helical structure. Complementary base pairs of A with T and G with C are formed by hydrogen bonding between the helices between the various nitrogenous bases. The base pairs are stacked perpendicular to the long axis of the double helix, 3.4 angstrom apart, with 10.5 base pairs per turn that spans 34 angstrom units. DNA can exist in several structural forms. Two variations of the Watson-Crick form are bDNA, or the A DNA and the Z DNA. Some sequence dependent structural variations cause bends in the DNA molecule. RNA also exists in several different single stranded structures, most of which are involved in protein synthesis. The linear array of nucleotides in RNA consists of adenine, guanine, cytosine and uracil, and the sugar moiety is ribose. Messenger RNA transfers the genetic information from DNA to ribosomes for protein synthesis. Transfer RNA and ribosomal RNA are also involved in protein synthesis. RNA can be structurally complex. Single RNA strands can be folded into hairpins, double-stranded regions or complex loops. Among the various several minor forms of RNA, ribozymes and siRNA have shown considerable attention from the scientists. Thank you. This is Dr. Padma signing off. Happy learning.